Now, what happens to the rest of the fatty acid? So in our fat mobilization, oops, let me start over again. In our fat mobilization, we had our triglyceride, and as we talked about, this is broken apart, forming our glycerol, which we just talked about how it's broken down for energy, as well as individual fatty acids. So, now we're going to focus on the fatty acids and talk about how they're broken down for energy, the catabolization of fatty acids. There are two steps for breaking down the energy. The first one is activation. The second one is beta oxidation. Activation, the fatty acid is converted to fatty acyl-CoA. This allows passage through the mitochondria membrane into the mitochondria. It's catalyzed by acyl-CoA synthetase. If you put synthase, I'd still give you credit. And keep in mind, as we're doing this, in order to activate it so that way we can release all of the future energy, we first need to energize it, aka prime it, aka activate it. To do this, we need to go ahead and energize it using available ATP. Now in this instance, usually we take off one phosphate and release energy. In this instance, we're going to release two phosphates, represented by PP. Now, when we take off two phosphates, it then converts our original ATP into a AMP, where M means mono. But this energizes our fatty acid as well as allows us to connect it up to a transport enzyme, which we call our coenzyme A. In this instance right here, the sulfur is going to go ahead and combine with the carbon, and then the alcohol and hydrogen are going to be released, and they form a brand new water molecule. This brand new combination of fatty acid and our coenzyme A, we call a fatty acyl coenzyme A fatty acyl coenzyme A. Now, throughout all of this, I'm going to refer to the rest as a fatty acyl coenzyme A. The reason for this is twofold. Technically, it's no longer a fatty acid because in order to have a fatty acid, you need a carboxylic acid. And that was removed when it was attached to a coenzyme A. So, oops, there's my sulfur, and then coenzyme A. So notice the alcohol was removed, and it's now attached to a sulfur group right here. So that's one reason why it's called a acyl. The other reason we call it an acyl is because eventually what's going to happen is that we're going to start oxidizing two carbons at a time. The end result is that the tail is going to be shrinking. So acyl is a generic term referring to the previous fatty acid that's going to be degraded over time, as well as attached to a coenzyme A. So notice the word fatty acyl. Spent a lot of time on that, I noticed. Once we've attached a coenzyme A, removed an alcohol, forming a fatty acyl coenzyme A, we then have beta oxidation. There are 4 steps in beta oxidation, and the first step I do want to point out to you, there's a typo. This right here should say, and I'll put this in red, dehydrogenation, because it forms a alkene from an alkane. So here are the steps: dehydrogenation, hydration, dehydrogenation, and finally release of a acetyl unit. So the whole goal in doing these steps is so that way we can form a brand new acetyl-CoA, which is then going to enter the Krebs cycle. The end result is that you'll then have a shortened fatty acyl group. In summary, and I'll keep in mind that's a D right here for dehydrogenation, so be sure to fix that. In summary, each pass through the fatty acid spiral produces one acetyl-CoA, one NADH, one FADH2. And I'm going to go ahead and explain the rest of this using the diagram on the next slide. Make sure all of this goes on your cheat sheet. So here are the steps: dehydrogenation, hydration, dehydrogenation, and release of a acetyl group. So first thing I want to do is show you what I mean by spiral. It turns out we're going to go ahead and, just as a piece of wood would have its outer coating of the wood charred and then taken away, and then the inner coating charred and burned and then taken away, releasing energy, and you get the point. Bottom line is, just as we degrade wood as we burn it, we're going to degrade the fatty acid. We're going to degrade them 2 carbons at a time. So go ahead and take a look at the very bottom. We're going to degrade it 2 carbons at a time. What happens is, as we degrade it, we're going to go ahead and release those 2 carbons in the form of acetyl coenzyme A. So let me draw my sulfur, coenzyme A. So we're going to release it in a form of acetyl coenzyme A. Because this is a repeating degrading process where we release 2 carbons at a time, we call this a death spiral, aka a spiral. Because what's happening is we're degrading it 2 carbons at a time. Whereas the Krebs cycle, where everything is constantly regenerated in a nice, perfect circle, in a spiral, we're degrading it. So what we're going to go ahead and do is focus on the first spiral. How do we sit there and release these first 2 carbons? These are the steps. We're going to first go ahead, use FAD, and regenerate it to FADH2. So we call this dehydrogenation, because we're going to be removing the hydrogens from these carbons right here. Now, this is going to be the tricky part. The carbon next to the carbon with the ketone we call a alpha carbon. Now, if we were to keep going to the left, the next carbon to that we call the beta carbon. So the other term for fatty acid oxidation is called beta oxidation. So FYI. So here we have the beta carbon. So let's go ahead and focus on the alpha and beta carbon as I explain this. Here on the alpha and beta carbon, we had 2 hydrogens taken off, and it formed a brand new alkene. We then have a hydration reaction, hydra for water. So the water is broken down, where the hydroxide goes on the beta carbon and the hydrogen goes on the alpha carbon. From here, we're going to go ahead and take off 2 more hydrogens. We take off the hydrogens on the beta carbon, where this hydrogen from the alcohol, this hydrogen from the carbon is taken off, creating a brand new NADH, and then this other hydrogen is just donated to the nearby solvent. So it's floating around on its own. So we then go from an alcohol to a ketone, and that's called the dehydrogenation step. In the last step right here, we have the release of the acetyl-CoA unit. So notice the whole goal is on the beta carbon to create a brand new ketone. With a brand new ketone here, we're then going to have a brand new coenzyme A. And it's going to go ahead and it's going to attach to this carbon right here. It then makes a brand new shortened fatty acyl group. So notice that's a new shortened fatty acyl group because the total number of carbons is going to be 16 carbons. Notice we originally started off with 18 carbons, but now it went to 16 carbons. So the other two carbons, notice, because it's going to go ahead and split as we add in the brand new coenzyme A to form the fatty acyl group. When we split it, we then released a brand new acetyl coenzyme A. Now this acetyl coenzyme A is going to go straight into the Krebs cycle. I'm just going to write KS, Krebs cycle. And eventually what's going to happen is we're going to have a leftover coenzyme A, which comes back from the Krebs cycle, and it's then just going to recycle itself. Now, this process is going to go ahead and repeat. So assuming that we took off these two carbons and we created a shorter a shorter fatty acyl CoA, we're then going to go ahead and repeat the steps. And it's going to keep going down the spiral till there's nothing left but a leftover acetyl CoA. So the whole purpose of the fatty acid oxidation is to go ahead and create all the acetyl CoA that we ever want. Know this information. I just talked about this earlier. They're energy dense, even though it's slow burning. They do have a lot of energy stores, so they're 25% more efficient energy stores than other biomolecules. I love this general information. So a total ATP from the 18 carbon generates a total of net 120 ATP. Keep in mind it did take 2 ATP to actually get this whole thing started. But as you can see, there was a lot more ATP that was produced in the long run due to all those acetyl CoAs that enter the Krebs cycle. So know this part, and know that it contains twice the amount of energy. So know those right there. I'm not going to test you on this other part right here, and let's move on. So let's go ahead and investigate starvation, or this also occurs in diabetes. There's an imbalance between carbohydrates and fatty acid metabolism. I just told you right now that in the fatty acid oxidation, the fatty acids are eventually reduced down to acetyl units, and then combined with acetyl-CoA. The acetyl-CoA then goes directly into the Krebs cycle. Here's the problem. If you are starving, you'll have a high amount of acetyl-CoA. But keep in mind, in order for this Krebs cycle to work, even though we're burning acetyl-CoA, we still have other biomolecules that are important. For example, we have oxaloacetate. It turns out that as you're starving, and that's a very messy starving, as you're starving, you have high amounts of the acetyl-CoA. However, you have low amounts of the oxaloacetate which is needed in the Krebs cycle. Why? Because the oxaloacetate is busy making brand new glucose to help keep the brain, the heart, your blood cells alive. So, in some ways, it's using the oxaloacetate to keep you alive, which is a good thing. Other ways, it's shooting itself in the foot, because then the Krebs cycle will no longer work. So what do you do with all this high amount of acetyl-CoA? Well, the body uses a temporary energy pathway, which we refer to as ketogenesis. And what it does is, and this only occurs when you're starving or if you're on what's called the Atkins diet. It can also occur during diabetes, but I'm not going to get into that one. Ketogenesis. So basically what happens is it's going to go ahead and use those abundant amount of acetyl groups, and it's going to turn them into what we call ketone bodies. The ketone bodies are then used as a energy carb substitute. Now, they're not as good as the original carbs, but at least they'll keep you alive. This process isn't as good as carbs, though, so you're not going to be able to perform any normal to athletic endeavors that you did when you ate normally. Furthermore, if your body has entered ketogenesis, it's already scrapping your proteins and muscle tissue and other parts of your body in order to make glucose, so you're actually still hurting yourself in the long run. But bottom line is, it will keep you alive if you're starving, and it will make use of all that body fat just to keep you going, at least for a couple weeks. So let's go ahead and explore this ketogenesis. In general, low carbs, less glycolysis, so less oxaloacetate for citric acid cycle, aka Krebs cycle, since the remaining amount is used in gluconeogenesis to form brand new carbs. So the bottom line is more acetyl-CoA produced by fatty acid oxidation that can be processed by the citric acid cycle. Nice new summary. So know this. The acetyl-CoA builds up, so ketogenesis occurs. Liver is very important because this is where it occurs in the liver. Converts the acetyl-CoA into keto bodies. What are the keto bodies? These are the ketone bodies. Make sure you know all of these: acetoacetate, beta-hydroxybutyrate, and acetone. I don't ask you to sketch it, so I wouldn't worry about it at this point. But bottom line is, it's these two right here. They are going to be used as the carb substitute. Keep in mind, this is not a foolproof process. This is a worst-case scenario for the body. So these molecules, when they enter the blood, it's very acidic. So there's various conditions. I'm just going to talk about the conditions briefly, not the long-term effects. But one way you know you're going into ketogenesis is because of ketonemia, where there's a high concentration of these ketones in the blood. And ketonuria, there's a high concentration of ketones in the urine. Consequently, your blood and urine can be a little bit, can make it acidic and acidify it. It's not going to make it like hydrochloric acid, but it's just one way that doctors can check for diabetes, since this is a side phenomenon. Another condition associated with ketogenesis to know that you're entering it is acetone breath. Your breath smells like that cleaning solvent we use to get rid of the organics out of our beakers, acetone. You have acetone breath. When all three happen at the same time, the body is said to have the condition called ketosis. Ketosis. So ketosis actually refers to these conditions. The actual chemical phenomenon to release energy from your fats is actually called ketogenesis. So make sure you notice those two differences. Let's go back to the first two being acidic. Ketoacidosis. So this can make your blood acidic, which is not healthy, and it inadvertently causes your kidneys to excrete large amounts of water to help counter it, so severe dehydration can occur. Now, the Atkins diet uses plenty of protein along with healthy fats, and there's a lot of arguments to and for the Atkins diet. I'm not going to get into that. What I will say is, even though it causes ketogenesis, if you want a sure, proven method to make sure you're healthy as well as has physical energy and contentment, I still say the best way is to have a healthy diet along with exercise, 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 exercise. And moving on. Now we're going to go ahead and get into fatty acid synthesis, but this is a good place to end this portion of the slides.